Hello everyone. Today we are going to talk about one of the very key concept of a chapter neural control and coordination, that is with respect to physiology of nerve impulse conduction. So I'm sure you have understood that nervous system and chemical system, that is endocrine system, together controls and coordinates various physiological activity of a body. Now we are going to understand how it does so. That means how our nervous system controls and coordinates a various physiological activity to maintain homeostasis in our body. You have understood the structure of neuron and various types and classification of neurons earlier, right? So neuron, which is a structural and functional unit of our body. In human body, we are having billions of neurons, and it is composed of two major components. There is cell body or a perikaryon, and number two, cell processes. Cell processes, which are again of two types, dendrites and axon. So today we will be understanding how axon and dendrites are carrying the impulse along its length. Not just that, न्यूरॉन के एक न्यूरॉन से दूसरे न्यूरॉन पे इंपल्स ट्रांसमिट कैसे होता है थ्रू साइनैप्स. So that is going to be our key discussion in today's class. So to understand how neurons conduct the impulse, we should first understand the key feature of neurylema, or a key significance of its plasma membrane, that is neurylema, because when we say that neurons are the excitable cells, यानी कि स्टिमुलस देने पे न्यूरॉन इंपल्स कंडक्ट करने की प्रॉपर्टी रखता है, दैट इज ऑल बिकॉज ऑफ इट्स नेचर एंड स्ट्रक्चर ऑफ न्यूरिलीमा. बिकॉज न्यूरिलीमा को हम रेस्टिंग स्टेट में पोलराइज्ड मेम्ब्रेन बोलते हैं. Now what do we mean by polarized membrane? That on either side of neurylema there is a difference in the charges. That is, outside of neurylema is said to be positively charged and inside of neurylema is negatively charged. ऐसा क्यों? What makes it positive, negative? Usually हम बोलते हैं सर ECF and ICF is equal number of positive and negative ions. I will not say equal amount of specific positive ion, but yes, ICF और ECF के अंदर पॉजिटिव नेगेटिव आयन का कॉन्सेंट्रेशन सेम होता है, बट न्यूरिलीमा के दोनों साइड पे अलग होता है. यानी कि आउटसाइड ऑफ न्यूरिलीमा हैज पॉजिटिवली चार्ज, इनसाइड ऑफ न्यूरिलीमा हैज नेगेटिवली चार्ज इन रेस्टिंग स्टेट. क्यों? बिकॉज न्यूरिलीमा shows presence of different types of channels, and these channels, which are of four major types, open and closes at a definite pattern, in a definite pattern. So when I talk about these channels, for me the most important are going to be these leak channels and voltage gated channels at this stage, to understand the impulse conduction along the axon and even across the synapse. But apart from that, chemical stimulation और मैकेनिकल स्टिमुलेशन के लिए भी न्यूरिलीमा के पास स्पेशलाइज्ड चैनल्स है. सो यस, व्हेन वी टॉक अबाउट दिस लीक चैनल्स, जो लीक चैनल्स है, यहां से आयन की मूवमेंट पैसिव फॉर्म में होती है. बिकॉज लीक चैनल आयन कॉन्सेंट्रेशन के हिसाब से ओपन या क्लोज होता है और वो मूवमेंट अलाउ करता है. इट इज सेड दैट न्यूरिलीमा के ऊपर potassium leak channels are more than that of the sodium leak channels. Neurylema के ऊपर potassium leak channels are more than the sodium. And when we talk about voltage gated channels, जिसको हम बोलेंगे, these are the most important role playing in case of nerve impulse conduction. Voltage gated channels specific potential difference पे ही open और close होते हैं. So yes, we will have major focus on these two channels, that is leak channels and voltage gated channels, and entire discussion of nerve impulse conduction. So look at this scenario. Outside of neurylema जहां पे सोडियम का कॉन्सेंट्रेशन ज्यादा है, इनसाइड ऑफ न्यूरिलीमा जहां पे पोटेशियम का कॉन्सेंट्रेशन ज्यादा है. ऑब्वियसली पॉइंट आता है सर दोनों तो पॉजिटिवली चार्ज आयंस है, तो दोनों जगह पॉजिटिव ही होना चाहिए, बट नो, अंडरस्टैंड दिस पार्ट. यहां पे ये नेगेटिवली चार्ज जो ऑर्गेनिक प्रोटीन्स जो है, दे आर कन्फाइंड टू ICF ओनली, यानी कि वो सेल के अंदर ही होते हैं, and that's the reason inside of neurylima becomes more negatively charged compared to outside, and that is a scenario in resting state, or this is why we call neurylima as polarized membrane. Okay, apart from that, neurylima के ऊपर के जो particular channels है, sodium channels, potassium channels, as I said, these leak channels are open and they allow free passive movement of ion as per their concentration gradient. As I said, potassium जो मेरा अंदर ज्यादा है वो इफ्लक्स दिखाएगा, सोडियम जो मेरा बाहर ज्यादा है वो इनफ्लक्स दिखाएगा. And as I said, potassium leak channels are more than sodium, so more positive charge ion is getting lost out compared to the positive charge ion coming in. So that also helps me in maintaining this resting potential difference, and that is about minus 70 millivolt. So during resting state, neurylima का resting potential जो होता है that is about minus 70 millivolt. Now, if I want neuron to conduct the impulse, I need to give stimulus. जैसे हम बोलते हैं अगर मुझे कार को स्टार्ट करनी है मुझे इग्निशन देना पड़ेगा, न्यूरॉन को मुझे चाहिए वो इंपल्स कंडक्ट करे, मुझे स्टिमुलस देना पड़ेगा. दैट स्टिमुलस, एज आई सेड अर्लियर, कैन बी केमिकल, मैकेनिकल और sensory or a physical, any form. So yes, when I give a stimulus, it has to have a minimum strength. That is what we call it as threshold stimulus. So एक minimum strength का जब मैं stimulus दूंगा तब जाके impulse conduction होगा. अगर मेरी stimulus की strength minimum से threshold से ज्यादा होगी तो भी speed of impulse conduction would remain same, और अगर उससे कम होगी obviously impulse generate होगी ही नहीं. So hyper threshold speed conduction remains same, hypo impulse generation will not take place. So let's understand what happens in this scenario. So let's say this is my neurylima. As I said, during resting state, inside is negatively charged. Whereas outside of neurylima is positively charged. That is because of outside having more amount of positive charge compared to the inside, right? And during this resting state, my resting potential difference is about minus 70 millivolt. So now when I give a threshold stimulus, when I apply a threshold stimulus जिसकी strength at least minus 55 millivolt होनी चाहिए, in the segment of the neuron जहां मैंने threshold stimulus apply किया है, sodium voltage gated channels will open. I repeat, on application of threshold stimulus, sodium voltage gated channels open. अब जब सोडियम वोल्टेज गेटेड चैनल ओपन होंगे, ऑब्वियसली सोडियम का कॉन्सेंट्रेशन जब बाहर ज्यादा है, इट विल शो इट्स इनवर्ड मूवमेंट, और व्हाट वी कॉल इट एज अ इनफ्लक्स ऑफ सोडियम. एंड दिस इनफ्लक्स ऑफ सोडियम generates action potential in that particular segment. यानी कि ये जो मेरा -70 मिलीवोल्ट था will move towards zero से वो जाएगा up to +30 to +40 millivolt. So this generates the action potential of +30 to +40 millivolt. This is all because of the influx of sodium. So can I say यहाँ पे inside of neurylima will now become positively charged, यानी कि मेरी polarity differ हुई, ये मेरा segment depolarize हुआ. So this basically leads to inside becoming positively charged outside will still remain negative, outside will become negative, whereas the remaining segment of the neurylema inside would be negative and outside is still positive. यानी कि मेरा ये जो segment है that has got depolarized, whereas rest of the segment is still in the polarized or a resting state. This is first step. Now, yahan action potential generate hua. Now, if you see inside only, there are difference in the charges. One segment is positive, other is negative. So this itself act as a stimulus for the next. यानी कि जब ये सेगमेंट में एक्शन पोटेंशियल जनरेट हुआ, अब सोडियम चैनल्स यहां क्लोज हो गए बिकॉज सोडियम वोल्टेज गेटेड चैनल्स विल ओपन ओनली इन दैट नेगेटिव रेंज ऑफ -55. अब जैसे ये पॉजिटिव की तरफ गया, यहां पे सोडियम वोल्टेज गेटेड चैनल्स क्लोज हो जाएंगे एंड व्हाट विल हैपन ओवर हियर, पोटेशियम वोल्टेज गेटेड चैनल्स ओपन होंगे. तो जब यहां भी पोटेशियम वोल्टेज गेटेड चैनल्स ओपन होगा तो क्या होगा? ऑब्वियसली सर, पहले तो पोटेशियम अंदर था, सोडियम भी अंदर चला गया, इनसाइड बिकेम पॉजिटिव. अब आप बोल रहे हो सर यहां पॉजिटिव के पोटेशियम के चैनल्स ओपन हो रहे हैं, पोटेशियम विल शो आउटवर्ड मूवमेंट. एट द सेम टाइम द नेक्स्ट सेगमेंट के सोडियम वोल्टेज गेटेड चैनल्स ओपन होंगे एंड दैट विल लीड टू इन्फ्लक्स ऑफ सोडियम. यानी कि in the first segment, sodium is still stuck in because of the closure of sodium voltage gated channels. Potassium will start moving out because of potassium voltage gated channels have opened because of the generation of action potential. And next segment में sodium will move in because of opening of sodium voltage gated channels. I repeat, generation of action potential in this segment. What it caused? It caused closure of sodium voltage gated channels, generation of action potential. What it leads to? Number one, closure of sodium voltage gated channels in that segment. Number two, opening of potassium voltage gated channels in the same segment. And number three, opens sodium voltage gated channels in the next segment. So yes, because of opening of potassium in that segment, there is an efflux of potassium, and opening of sodium in the next segment, there is the influx of sodium. So over here, in this segment 2, potassium was already in, sodium is also going in. So outside will now become negative and inside will now become positive. Whereas over here, as the potassium is coming out, more positive charge is coming out, outside will again become positive and inside will become negative. यानी कि अब ये सेगमेंट डीपोलराइज हुआ और यहां पे पोलैरिटी वापस से रिवर्स हो गई, ये सेगमेंट रीपोलराइज हुआ एंड यहां पे जो आगे का सेगमेंट है वो तो अभी भी रेस्टिंग स्टेट में है, यानी कि ये अभी भी पोलराइज्ड स्टेट में है. यहाँ पे inside potassium is there and outside sodium is there. This is... So now the question comes, sir क्या ये resting state और repolarized state को same नहीं बोला जा सकता? The answer is yes as well as no. Yes, because polarity wise resting and repolarized दोनों state में outside is positive, inside is negative. और no इसके लिए ionic distribution. अगर हम आयोनिक डिस्ट्रीब्यूशन देखते हैं तो रीपोलराइज स्टेट में पोटेशियम बाहर है, सोडियम अंदर है, और पोलराइज यानी कि रेस्टिंग स्टेट में सोडियम बाहर है, पोटेशियम अंदर है. मतलब depolarization or generation of action potential of this segment, I would label it as B, will act as a stimulus for C. यानी कि अब बी में सोडियम वोल्टेज गेटेड चैनल्स क्लोज हो गए, पोटेशियम ओपन हुए, एंड द सेम सिनेरियो सोडियम वोल्टेज गेटेड चैनल्स ऑफ सी विल ओपन. सो होगा क्या, अब यहां पे हमें सोडियम की इनवर्ड मूवमेंट दिखेगी, यानी कि ये सेगमेंट डीपोलराइज होगा. यहां पे हमें पोटेशियम की आउटवर्ड मूवमेंट दिखेगी, यानी कि ये सेगमेंट रीपोलराइज होगा. सर इसका क्या, यहां पे रोल आएगा सोडियम पोटेशियम पंप का, और आई वुड से सोडियम पोटेशियम एटीपी पंप का. सो लेट अस अंडरस्टैंड इन अ लिटिल बिट मोर डिटेल. यानी कि अब हम वापस एबीसी पे आते हैं. सो ऑब्वियसली इन दिस सेगमेंट sodium voltage gated channels have opened, so influx of sodium, potassium was already inside has become positive, inside has become positive, outside will remain negative or become negative. Whereas in B segment में क्या हुआ है, efflux of potassium, sodium was stuck inside has become negative and outside will become positive. Whereas in A segment, as I said, comes the role of sodium potassium pump, which will throw out 3 sodium molecule for entry of every 2 potassium molecule. And that's how in this A segment, inside will again remain negative and outside, because more positive charge are going out, outside will become positive. That means now this segment has got depolarized. This segment, because of the efflux of potassium, repolarized, and this segment has become polarized because of the sodium potassium ATP pump. Repolarization due to efflux of potassium, and depolarization due to influx of sodium. So this is how impulse will travel along the neurulema from one segment to another. Right? अगर हम NCERT को थोड़ा सा यहाँ पे recap कर ले, this is a picture which you have in your 11th grade NCERT, where initially we spoke about depolarization, where the stimulus has been given, and then the positive charge moves on to the next segment. Yani ki this act as action potential for the next, this act as a stimulus for the next. This is how it will keep moving. Right? A little more pictorial form for a recap. Yes. Starting from here, during the resting state, this is how sodium channel is closed, potassium channel is also closed. Voltage gated channels, as we said, voltage gated channels open and close only at a specific voltage. Now, as the threshold stimulus has been given, you brought -70 to -55, sodium voltage gated channels opens, so sodium moves in. Right? Next, where more and more sodium coming in, inside became positive, outside became negative. So, generation of action potential, sodium channel closed, potassium open. Over here, potassium channel open. And as soon as the potassium channels open, positive charge or potassium charge is moving outside of neurulema again regains its polarity. So repolarization, and polarity wise, it has coming back to its initial state, that is outside positive, inside negative. Right? So this is how resting state, threshold stimulus, generation of action potential. So I would say number 3 over here, denotes influx of sodium leading to generation of action potential. Number 4, that denotes efflux of potassium leading to depolarization. At times, more potassium goes out leading to hyperpolarization. But as I said, more potassium leak channels are there. तो अगर potassium बाहर ज्यादा भी चला गया, वो leak channel के through वापस अंदर आ जाएगा and it will maintain back to the resting state. Right? And अगर पंप की बात कर रहे हैं, एटीपी सोडियम पोटेशियम पंप की बात कर रहे हैं, yes, look at the structure. At times we say, sir, why 3 sodium are going out and only 2 potassium are coming in? That's the structure of the sodium potassium pump. मतलब सोडियम पोटेशियम पंप का स्ट्रक्चर इस तरीके से बना है कि 3 सोडियम मॉलिक्यूल यहां पे अटैच हुए, वो अंदर की तरफ आए, और 3 सोडियम मॉलिक्यूल जो पंप पे अंदर से अटैच हुए बाहर की तरफ गए, 3 सोडियम मॉलिक्यूल जो अटैच हुए बाहर की तरफ गए, और 2 पोटेशियम अंदर की तरफ आए. That's how more and more positive charge is going out compared to the charge coming in. Right? So this is how neurylema is brought back to its original polarized or a resting state. Okay. So ये तो हमारी बात हुई how the nerve impulse is conducted along the non-myelinated nerve fiber, where impulse has to travel the entire distance of the or entire length of the nerve fiber. But what happens in myelinated nerve fiber? Because when we look at the myelinated nerve fiber, where Schwann cells or oligodendrocytes have secreted the myelin sheath, which is not a continuous sheath. Again, understand this, one of the very important phenomena. Myelin sheath, which is made up of lipid, white in color, is not a continuous sheath, but it shows discontinuation, and those discontinuation or junctions are called as nodes of Ranvier. Okay, so these channels are there along the entire length of the neurylema, but the part of the neurylema which is covered by myelin sheath does not allow ion exchange, and the impulse or action potential is generated only from node of Ranvier to another node of Ranvier. So here impulse jumps from one node of Ranvier to another node of Ranvier, and that's why to jump meaning salter, and that's why myelinated nerve fiber में जो impulse conduction होता है उसको हम बोलते हैं saltatory impulse conduction. We call it as saltatory nerve impulse conduction, and that's where the speed of impulse conduction over here is about 100 to 120 meter per second. Whereas myelinated nerve fiber mein, as the impulse has to travel the entire length, the speed is about 15 to 20 meter per second. Over here it is about 100 to 120 meter per second. Now next is about how the impulse is carried across the synapse. Throughout the length of the nerve fiber, it is electrical. That impulse conduction is electrical. Ion exchange के form में हो रहा है. Synapse में कैसे form में होता है? That across the synapse, the impulse conduction is in the form of chemical, and that's what we call it as neurotransmitter coming into play. So let us understand the structure of synapse first, and then we'll understand how this neurotransmitter helps in transmission of impulse across the synapse. So when I look at the structure of synapse, let's say this is my nerve ending, or what we call it as an axonal terminal or a telodendrons, and here I have a next neuron, उसके dendrites. So when I magnify this part, what I can see, a synaptic knob or a synaptic bulb contains vesicle, fluid-filled vesicles. What we call it as synaptic vesicles. vesicles. And these synaptic vesicles are the one which are having the neurotransmitter. Neurotransmitter is the one which is synthesized by Niesel's granule, which are present only and only. Niesel's granules are present only in cyton and dendrites. Important MCQ point. Exons में Niesel's granules नहीं होते, तो ये synaptic vesicles dendrites और cyton से बनके टीरोडेंड्रॉन में synaptic knob में यहाँ आके स्टोर होते हैं. So this synaptic vesicles containing neurotransmitter. Now this membrane, we call it as presynaptic membrane. नेक्स्ट न्यूरॉन का जो मेम्ब्रेन है उसको हम बोलेंगे पोस्ट साइनेप्टिक मेम्ब्रेन. और ये जो गैप है, दिस गैप is what we call it as synaptic cleft. Okay, so when the impulse reaches to the synaptic knob or a synaptic bulb, there is an influx of calcium. There is an influx of calcium, and this calcium ion diffuses the synaptic vesicle with the presynaptic bulb, presynaptic membrane, and all this secretion fills the gap, fills the cleft. And this filling of the cleft stimulates these channels present on postsynaptic membrane to allow influx of sodium, leading to depolarization of the next neuron. I repeat, as the impulse reaches to the synaptic knob, influx of calcium, this calcium diffuses synaptic vesicle with the presynaptic membrane. Presynaptic membrane allows this diffusion of neurotransmitter, and that neurotransmitter stimulates this voltage gated channels of the next neuron, allowing depolarization. Okay, so that's the reason impulse conduction in higher animals, is called as electrochemical. That along the length of the neuron, it is electrical. Across the synapse, it is chemical. In lower animals or in certain fishes, we have seen that synaptic cleft is so narrow, from one axon, directly electrical impulse goes to the next. Right? So, वहाँ पे सिर्फ electrical है. वहाँ पे bidirectional हो सकता है. But in human, on higher animals, nerve impulse conduction is unidirectional. क्यों unidirectional? Dendrite के पास केमिकल रिलीज करने की एबिलिटी नहीं है. डेंड्राइट के पास न्यूरोट्रांसमीटर नहीं है. डेंड्राइट्स पे न्यूरोट्रांसमीटर एक्ट करते हैं. डेंड्राइट्स न्यूरोट्रांसमीटर सीक्रेट नहीं कर सकता. दैट्स द रीजन इन हायर एनिमल्स impulse conduction is unidirectional, whereas electrical impulse transmission can be bidirectional as well. So that's all with respect to the physiology of nerve impulse conduction for your NEET preparation. All the best.